Hello dear students, today I am going to deliver a lecture on the topic Colletotrichum, important features and life history. Main objectives of this lecture are to make students aware about the economic importance of Colletotrichum, to highlight important morphological and reproductive features of Colletotrichum, to study the life cycle of Colletotrichum. Colletotrichum is a genus of deuteromatous fungi. Several plant pathogenic species have been recognized in this genus. They also infect some animals. Members of this genus cause anthracnose diseases of cereals, grasses, legumes, vegetables, and fruits. Not only are they plant parasites, but they also infect animals. Biotransformations employing Colletotrichum species is also a growing area of research nowadays. Mycelium of Colletotrichum species is internal and septate. It grows inter as well as intracellularly. Conidiophores arise from the reproductive structures called acervulus and gives rise to conidia to continue the life cycle asexually. Now, introduction to this genus. Colletotrichum is a genus of deuteromatous fungi belonging to class Coelomycetes. The genus Colletotrichum was first described by Todd in 1790 under the name Vermicularia, but was later established as Colletotrichum by Corda in 1837. Von Erx in 1970 has recognized 11 species in this genus, which are parasitic on different plants. Colletotrichum is one of the most common and important genera of plant pathogenic fungi. Colletotrichum species cause typical disease symptoms known as anthracnose. The word anthracnose comes from the Greek word anthrax, meaning carbuncle, which also means an abscess larger than a boil, and nos, which means disease. Anthracnose is characterized by the production of spore-bearing body called an acervulus, which usually looks like a tiny black spot on the host surface. Anthracnose disease attacks all plant parts at any stage. The symptoms can occur on the leaves and ripe fruits. In the beginning of the disease, anthracnose generally appears on leaves as small and irregular yellow, brown, dark brown, or black spots. The spots expand later and combine to cover the whole affected area. The color of the infected part darkens with the age. The disease can also produce cankers on the petioles and on stem that causes severe defoliation and rotting of fruits and roots. Infected fruit has small, water-soaked, sunken, circular spots. As it ages, the center of an older spot becomes blackish and emits gelatinous pink spore masses. Virtually every crop grown throughout the world is susceptible to one or more species of Colletotrichum. Colletotrichum fungi cause post-harvest rots, anthracnose spots, and blights of aerial plant parts. Members of this genus cause major economic losses, especially of fruits, vegetables, and ornamentals. Not only are they plant parasites, but they also infect animals. Several species have been reported to be opportunistic pathogens of humans. Colletotrichum truncatum was isolated from a corneal ulcer of a human eye. Colletotrichum dementium was also reported to cause eye keratitis, a potential ocular disease. Colletotrichum fungi are highly significant as experimental models for studying many aspects of fungal development, infection process, host resistance, signal transduction, and molecular biology of plant pathogen interactions. Now, vegetative structure of Colletotrichum. Colletotrichum encompasses species with endophytic, epiphytic, saprobic, and phytopathogenic lifestyles. The mycelium is endophytic, that is internal, septate and branched. Hypha grow both intercellularly and intracellularly. Colletotrichum species have been found as symptomless inhabitants, that is endophytes, in plant tissues. Colletotrichum strains were isolated from healthy leaves and pseudocystems of banana. Hyde and Soitog in 2008 discussed the role of endophytes that can become primary saprobic decomposers and act as latent infectious pathogens that cause diseases under specific conditions. In Colletotrichum falcatum, the mycelium is found in the parenchymatous cells of the pith. In case of Colletotrichum lindemuthaneum, which causes anthracnose of bean, the mycelium is localized in the lesions only. In the beginning, mycelium is hyaline but turns dark at maturity. Cell contents also become denser and thicker with the age of the hyphae. In the host plant, the mycelium develops first in the epidermal cells and later grows as inter- and intracellular hyphae in the cells of the host. The thick-walled hyphae finally collect beneath the cuticle and become compacted to form a black structure called stroma. From the surface of the stroma, clusters of black velvety bodies called acervulus are formed. Each acervulus consists of a stromic layer from the surface of which arise hyaline and unseptate conidiophores. Now, reproduction and life cycle of Colletotrichum. The only method of reproduction is asexual reproduction. It occurs by means of conidia. Conidia are formed in asexual fructifications called acervuli. An acervulus appears as a saucer-shaped structure and is surrounded by stiff, black, unbranched hairs or bristles termed as setae. These setae are septate. From the upper surface of the stroma, inside the acervulus, arise short, one-celled branches called phallates or conidiogenous cells or conidiophores. Acervuli may be subcuticle, subepidermal or epidermal with a characteristic peripheral ring of black, long and stiff setae. From the tip of a conidiophore, the conidia are formed acrogenously by abstraction. Each conidium contains an oil globule and is aseptate and sickle-shaped. With the maturation of the conidia, the host epidermis gets ruptured. The life cycle of Colletotrichum species begins when spores land on host surface. Then a cascade of events like spore germination, germ tube formation, and aprosorium development starts. Conidia are dispersed by wind or rain. Moisture is essential for conidia formation and germination. The spores form one or more germ tubes which form new mycelium. An aprosorium is formed when the germ tube comes in contact with the host surface. The infection peg or penetration hypha penetrate the cuticle at the contact of epidermis and aprosoria. At this place, hypha swells to form a vesicle and a broad primary hypha, which are surrounded by invaginated plasma membrane. The host protoplast remains alive during the biotrophic stage. Interfacial matrix separates the protoplasts of fungus and host. 1 or 2 days after penetration, plant plasma membrane disintegration starts. As new host cells are colonized by primary hypha, the sequence of a transient biotrophic phase followed by cell killing is repeated. This leads to growth of fungi internally. Sometimes in the old mycelium, thick-walled dark brown angular cells develop terminally or at intercalary position. These cells develop oil within them. When they detach, they germinate on the substratum to form the new mycelium. They are known as chlamydospores, which serve as perinating spores, since conidia cannot remain viable for a long time. Thus, intracellular hemibiotropy is a distinctive aspect of Colletotrichum pathology. Following penetration of the host epidermis, Colletotrichum species initially colonize living plant cells. After some time, the growth habit switches to necrotrophy and massive cell death and tissue destruction starts. This switchover in the lifestyle is associated with dramatic and well-characterized morphological, genetic, and physiological changes in the hypha. Colletotrichum genus have been cloned that are differentially expressed during the 2 phases. Many Colletotrichum species have the capacity to produce quiescent infections on various plants. Many Colletotrichum fungi are also endophytic on their host plants. Thus, Colletotrichum presents a unique blend of mixed lifestyles, namely biotrophy, necrotrophy, endophytic, and latent mode within a single genus and within a single species. Mycelium of the Colletotrichum is internal and septate. It grows inter as well as intracellularly. Conidiophores arise from the saucer-shaped structures called acervulus and give rise to conidia to continue the life cycle asexually. Morphological characters such as shape and size of conidia, setia, and aprisoria have been used to characterize various Colletotrichum species. Reproductive structures are key to delimit this genre. The most significant advances in taxonomy of this genre have been obtained from various approaches based on the analysis of nucleic acids like restriction, fragment length, polymorphism, that is RFLPs. Now, epidemiology of diseases caused by Colletotrichum. Environmental factors play a major role in the development of Colletotrichum diseases. Colletotrichum affecting legumes develop best at a temperature of 18 to 28 degrees and a relative humidity of greater than 90%. In general, the primary factor influencing the development of disease is the availability of water, as high humidity is essential for the sporulation of most of the Colletotrichum species. Other key factors such as temperature often interact with factors such as leaf surface wetness and humidity, mist or dew, thus playing a key role in disease development. Now, effect of Colletotrichum on agricultural crops. Colletotrichum species have been reported on a wide range of hosts. However, the most prominent of these are species attacking cash crops such as cotton, coffee, beans, and tropical fruits like banana, mango, and papaya. Losses caused by the Colletotrichum species mostly occur as a result of the direct reduction of quality and quantity of the harvested yield. The disease is most noticeable and severe when Colletotrichum directly attacks the harvested portion of the crop, often the fruit. Wider economic consequences are also linked with other factors, including the poor return on investment and the cost of chemical control. Many export crops play a crucial role in the economy of small developing countries, and disease losses can have a significant impact at the macroeconomic level. At the microeconomic level, reductions in the yield losses have a significant effect on the lives of people, many of whom are small marginal farmers or dependent on local markets for food. Some important pathogens of this genus are Colletotrichum falciparum on the sugarcane, Colletotrichum legionarium on cucurbits, Colletotrichum gossypi on the cotton, Colletotrichum graminicola on the seeds of many grasses and cereals, Colletotrichum musarum causing the anthracnose and fruit rot, also called cigar ant disease of banana. The above ground plant parts can be affected by Colletotrichum diseases at all stages on the stems, leaves, flowers and fruits. Colletotrichum kawai causes anthracnose in coffee berry. Colletotrichum acutinum parasites strawberry. Colletotrichum orbiculari causes anthracnose in watermelon, while as Colletotrichum lindemethanium causes bean anthracnose. Black dot of potato is caused by Colletotrichum coccodus. Anthracnose leaf blight of maize is caused by Colletotrichum graminicola. Now, description of some important plant diseases caused by this genus. Anthracnose of turfgrass. Anthracnose of turfgrass is caused by Colletotrichum graminicola, also called Colletotrichum cereali. A number of turfgrass species are host to this pathogen, but anthracnose is more common disease of annual bluegrass, Poa annua, and bent grass agrotis species. Symptoms of this disease. It is a foliar disease. Foliar lesions appear reddish in color. The foliar phase produces elongated chlorotic leaf spots and black acervuli. Finally, leaves turn brown. Black setae on plant surface are key sign of this disease. It makes large fields of bent grass patchy. Lawns and golf courses affected by this pathogen give a patchy look. Now, disease cycle of this pathogen. Spores are produced in acervuli, and these are water splashed onto crowns and foliage. The spores germinate, produce aprosoria. Aprosoria serve to anchor the fungal filaments to the plant surface and directly penetrate into the plant epidermal cells. A penetration peg emerges from a pore in the aprosorium and penetrates plant epidermal cells directly. Penetration of the epidermis is closely followed by production of a single irregularly shaped infection hypha. One or more branches from the infection hypha then invade the host. Colletotrichum cereali behaves as a necrotroph, killing plant tissues with enzymes and possibly surrounding plant mesophyll cells. The sexual stage, that is telomorph, of Colletotrichum cereali is the ascomycete glomerula graminiculae. The fungus feeds initially without visible damage, but then plant cells start to die and collapse as the fungus grows from cell to cell. After that, the fungus produces acervuli on the surface of the infected area, from which spores are released to start the life cycle again. Colletotrichum cereali overwinters as mycelium or conidia associated with previously infected plant tissues. Now, another disease is black dot disease of potato. Black dot disease of potato is caused by Colletotrichum coccodus and is a common disease of potato. It is most often observed on tubers but can affect all parts of the plant. It can be more devastating as it affects all parts of the plant. It causes wilt in above ground parts and severe rotting of roots and shoots. Discoloring of tubers is also a common phenomenon, thus reducing the yield. Black dot is named after the abundant black dots on tubers. The black dots are microsclerotia that are often visible to the naked eye. Black spots are found on stolons, roots and stems. Stem lesions are more common. Stem lesions tend to form around the base of the leaf petiole. As with leaf lesions, they initially start as small brown flecks. These gradually combine, forming lesions which may girdle the stem. Lesions on maturity develop circular to irregularly shaped white to straw-colored centers with wide margins that vary in color from brown to black. Microsclerotia form in the center of the lesions and are often clearly visible against pale background. As infected tissue shows signs of senescence, microsclerotia may become abundant, covering the entire surface of the stem. Tuber symptoms appear as a brownish to gray discoloration over a large portion of the tuber or as circular to irregularly shaped areas. Disease cycle. Colletotrichum coccodus readily produces microsclerotia on the infected plant tissues and the surface of the tubers. These structures allow the pathogen to survive for long periods of time in the soil. The pathogen survives between growing season as a sclerotia in infested plant debris and infected potato tubers. It also overwinters in the debris of susceptible solanaceous crops and weeds. In the spring, sclerotia develop into acervuli. Spores serve as the primary inoculum to initiate the new disease. Inoculum is spread through water and air. Colletotrichum coccodus grows best at temperatures above 68 degrees Fahrenheit and free moisture from rain, irrigation, fog or dew are required for spore germination and infection of new plant tissues. Spores land on susceptible plant tissue. They germinate and may penetrate tissues directly through the epidermis or through the wounds. Infection of above ground plant parts continues throughout the growing season, especially when plants are under stress, such as during long, warm, dry periods. Production of microsclerotia is greatest at high temperatures, and increasing disease incidence has been associated with increasing soil temperatures. Colonization of tubers can occur at all stages of development, but formation of sclerotia on the tuber surface is more prevalent late in the season when soil temperatures are at their highest. Now another disease caused by this Colletotrichum is coffee berry disease. Causative organism for this disease is Colletotrichum cavai. Symptoms of this disease are: this pathogen infects all stages of the coffee crop, from flowers to ripe fruits and occasionally leaves. Maximum crop losses occur following infection of green berries. It leads to the formation of dark, sunken lesions with sporulating acervuli, causing premature drooping and mummification of berries. It may cause up to 70 to 80% losses if no control measures are adopted. Now, disease cycle of this pathogen. The early stages of fungal development on the host plant surface are germination of conidia on host to produce germ tubes, which later on form aprisoria for penetrating the cuticle directly. Fungal penetration into the host may be based on mechanical pressure exerted by melanized Eprosoria, the secretion of cutin degrading enzymes, or by a combination of both the processes. Penetration of different host parts like hypocotyl, leaves, and green berries also occurs directly through the epidermal cell walls. Now another disease caused by Colletotrichum is red rot disease of sugarcane. This disease is caused by Colletotrichum falcatum. The disease is prominent in subtropical areas of the world. It is also common in India. It affects stems, leaves of the sugarcane. As soon as sugar formation starts, symptoms begin to appear. Now, symptoms on leaves. Loss of turgidity and discoloration of leaves is one of the main symptoms. Leaves droop very soon, thus transpiration and photosynthesis are reduced. In the late stage, oblong, blood red necrotic lesions appear all along the midrib. In the older lesions, black velvety acervuli may appear. When the conidia appear on the leaves, they get covered by a powdery mass. Now symptoms on the stem. The rind loses its color and splits up lengthwise. Canes become internally rotted. The pith becomes red and is exposed. Vascular tissues also turn red. Thus it is called red rot. Later in the season, acervuli are formed as small velvety dark areas on the canes. The splitted stem emits alcoholic smell due to the fermentation of sucrose into alcohol by pathogenic enzymes. Now the disease cycle of Colletotrichum falcatum. After infecting the host, this pathogen spreads rapidly. It forms cylindrical, branched, septate, colorless hypha. Masses of hypha can grow both intracellularly as well as intercellularly. Hyphal infection reaches up to pith and forms chlamydospores in the pith, which perinate in the soil. Hyphal masses also form acervuli. Later, conidiophores develop on the acervuli. The former develop conidia at their tips. Conidia are falcate in shape, hyaline and one-celled. Conidia are dispersed by wind, water and insects. They reinfect new cells of the host. Infected seed sets are the main means of annual recurrence of the red rot. Predisposing factors of this disease. Several factors are responsible for the perpetuation of this disease. Waterlogged soil and high humidity make the disease very dangerous. Growing a single species for several years also causes red rot recurrence because of the inoculum development over successive years. Dear students, I hope you have now fully understood this topic. Wish you success. Thanks.